Okay, so we're going to cover Lab 12 special senses, and I'm going to toggle a little bit back and forth with the actual handout sheet that we're going to fill in the answers and discuss it. We will go through maybe a few of the images, that are in the image database there. And if you look below that region, I have links to videos that I actually physically cover each of the models. So those are very, very helpful. So below where you can have all the little lab images, 12-1, 12-2, 12-3, below that, it'll say ear model, cochlea model, eye model, and those are individual Zooms, and recordings, videos of me covering the anatomy, and their functions. Okay, so let me switch to the document here. I might need to download it, but I think it's going to be fine like this. Okay, so here we have exercise 1, the anatomy of the eye and vision models. So again, there are models to correspond with this. We'll go back through those and identify what's the cornea, the iris, the pupil, so forth, the different muscles and so forth. So at this point, all we need to do is match up the functions. All right, so let me move this thing up above a little bit. So what I do, and almost always do, is I start with the option like A, the group of muscles which move the move or roll the eyeball, and I then go up to the list and just match it. You can write the letter in there for now, and then copy the function into the space provided on the right, however you want to do it. All right. So we're starting with the group of muscles that move or roll the eye. Those would be the rectus muscles there, superior, inferior, medial, and lateral rectus, and superior and inferior oblique muscles. All right, so A goes to the rectus muscles. B, the connective tissue that gives the eye shape. Right? That would be the sclera. Then we have C, the anterior clear covering that allows light to enter the eye and refracts light. That would be the cornea. D, the highly vascular layer that absorbs light. That would be the choroid. E, secretes aqueous humor and contains the ciliary muscle which alters lens shape. That would be the ciliary body. F, the colored portion of the eye that regulates the amount of light entering the eye. The colored portion is the iris. G is the hole in the center of the iris. That would be the pupil. H, the layer that contains photoreceptor cells, as well as other cells there, like the bipolar cells and the ganglion cells. That would be the retina. I, the cranial nerve that transmits visual stimuli to the brain. That's the optic nerve. J, the biconvex structure that functions to refract light and bend it so it gets focused on the central fovea or the fovea centralis. That would be the lens. So J is the lens. K, the fluid in the anterior region that nourishes the lens and the cornea. That is the aqueous humor. L, the non-replenished fluid in the posterior region that maintains eye shape and holds the retina in place. L is the vitreous humor. M, the area of high visual acuity and cones. That would be the central fovea or the fovea centralis. And N, secretes tears to lubricate and cleanse the eye. That would be the lacrimal gland. Okay, that takes us to the next exercise, the extrinsic muscles of the eye and cranial nerve innervation. So it's here, right, that we match the cranial nerve, that we studied last week, that supplies the muscle. So there's an easy way to keep this in mind. We always used it, when I was in school, LR6, SO4, all else 3. Which means superior oblique is innervated by cranial nerve 6, which is abducens. SO4 superior, SO4, I'm sorry, I did it the opposite way. SO4, the superior oblique is innervated by trochlea. LR6, blah, lateral rectus is innervated by abducens. And all the other muscles are innervated by oculomotor. All right, so LR6, SO4, all else 3. Okay, so superior rectus is innervated by cranial nerve 3, or you can write 3 in there, or oculomotor. Inferior rectus, cranial nerve 3, oculomotor. Medial rectus, cranial nerve 3, oculomotor. Lateral rectus, cranial nerve 6, abducens. Superior oblique, cranial nerve 4, trochlear. And inferior oblique, cranial nerve 3, oculomotor. So when we look at the models, you need to be able to identify which muscle is which, okay? Superior rectus, inferior rectus, superior oblique. But you also need to know the nerve that supplies it. Okay, then we get to the microanatomy of the eye slide. So when we're looking at the slide of the retina, and that's what the other video was for, you know, we had a live online Zoom, and that Zoom, it's hard to do in the classroom. The only benefit that came from that is that I could have the slides on the screen and I could actually talk and point to them and have that recorded like I did with the other models, you know, for the eye and the ear and so forth, you know, but there was an open question and answer at the beginning that was rather extensive. So if it's possible, I will try to resume that, you know, without having the class being able to get in, you know, and I'll just cover those slides like I'm doing right now from my office, but do it in the lab. So it's synopsis. But we can also, I believe there's a picture of it in the lab images that Dr. Brightman uploaded, you know, so let me go back through there after we're done with all this. And if there isn't, then I'll, you know, I'll do another Zoom that's briefer or more brief, I guess you should say. Okay, so when we're looking at that, you should be able to see the sclera and the choroid in the retina, right? The sclera would be the outer connective tissue, kind of the white part. The choroid looks like it's dried blood area because it's a highly vascular area. And so when the slide is prepared and the tissue is obtained, the blood kind of congeals and dries, and so it comes like kind of like a black color. And then this rather thick region is the retina, and it has 3 layers of cells. So the layer, you know, the structures would be the sclera, choroid, and the retina, and the retina can then be broken down into the 3 layers of cells: the photoreceptor cell layer, the bipolar cells, and the ganglion cells. So if we looked at the sclera and then the choroid, the retina would be next. The first layer of cells, if you were working from kind of superficial to deep, from sclera to choroid, you know, the first layer of cells would be the photoreceptor cell layer, then the bipolar cells, and the ganglion cells. And the ganglion cells are essentially the most medial, you know, or deep of all these. That would be kind of intimate with the vitreous humor, the fluid that's found in the posterior chamber. All right, so we'll take a look at those as best as we can. Okay, then we go on to the anatomy of the ear, hearing, and equilibrium. So again, structure, and function, right? We have the pinna, external auditory canal or meatus, tympanic membrane, our ear ossicles, which are 3 small bones, malleus, incus, and stapes. The eustachian tube, now the textbooks call it the pharyngotympanic tube because it connects the upper part of the throat, called the nasopharynx, to behind the tympanic membrane. But frequently it's still referred to as the eustachian tube, but you should be familiar with both terms. The cochlea to semicircular canals, the hair cells of the organa corti, and the vestibulocochlear nerve, right? So those are the only things we're going to identify on those models, right? There are some other structures like the vestibule, you know, that is associated with static equilibrium. It's kind of a almond egg-shaped area between the semicircular canals and the cochlea in the inner ear. You know, but not all the models have numbers or arrows pointing to them. So I'm not going to require you to identify it on a model. All right, so we're going to go to the functions as we would have before. Right. We have the bony spiral canal that contains the hearing receptors. So A, the bony spiral canal that contains the hearing receptors is the cochlea. B, the hearing receptors is the hair cells of the organ of Corti. C, conducts vibrations through the middle ear. That would be the ear ossicles, malleus, incus, and stapes. D, transmits sound waves to the tympanic membrane. It also is the area where we secrete earwax to prevent material from entering the ear. I mean, it actually from coming in contact with the tympanic membrane, it's never going to get across that membrane unless the tympanic membrane is perforated. So D would be the external auditory canal or meatus. E equalizes pressure between the middle ear and the atmosphere. That would be the eustachian tube or the pharyngotympanic tube. F conducts action potentials to the brain. That, of course, is going to be the vestibulocochlear nerve. G funnels sound waves into the ear. That would be the pinna or the auricle. And I, equilibrium, dynamic equilibrium. That would be the semicircular canals. All right, so I would be the semicircular canals. In lecture, hopefully you've seen the pathway and sort of how these sound waves that are kind of bouncing around, outside of your head, need to be funneled, just like a funnel in cooking or in how you might put oil in your, change the oil and put some more oil into your engine, is that you use a funnel, and your ears or your pinna are kind of half funnels, and they capture the sound waves and they direct them down your external auditory meatus or canal. And so it kind of gets congregated and funneled to hit that tympanic membrane, put the ear ossicles in the motion, the oval window, the fluid in the cochlea, so that we can stimulate the hair cells of the organ of Corti. So it makes it more efficient. It's somewhat similar to the cornea and the lens trying to refract and bend light photons so that they hit optimally and they converge right on the center of that fovea centralis, because you have this high population of cones there for color. And if we can get it to, target that one spot, you have very accurate vision. Okay, so then we go to the cochlea anatomy. There's a large model where we're going to identify the scaba vestibuli, the scala tympani, the cochlear duct, the cochlear branch of the vestibulocochlear nerve, the vestibular membrane, the tectorial membrane, and the hair cells of the organ of Corti. Then my video on that is very good. Sometimes I did the videos on the models because there weren't real good pictures already on the course page. Okay, and then we move on to taste buds. All right, so the sense of taste or gustation is detected by these gustatory cells in the taste bud. And the taste buds are found in the stratified squamous epithelial layer, or the kind of like what would be similar to the epidermis of the skin, is in that surface area. So we have these taste buds. They kind of look like a grapefruit that's been peeled, where you can see large sections, of the grapefruit, or maybe a large orange. Sorry, everything reminds me of food. And so they have that sort of shape to them. They expose themselves, between the taste bud and the epithelium, there's a pore, so that as food is chewed and macerated and mixes with saliva, now those tastants, or the molecules in the food, become suspended in the aqueous portion, the watery portion of saliva. And as that food passes across the tongue, it will fall into these crevices or papillae, and the taste buds open up to that crevice. So where we have these taste buds, it's sort of like the cerebrum where you have gyri and sulci, it's going to undulate. And where you have the crevice there, the taste buds will open up to that, interior region, and it gives them an opportunity to come in contact and detect the presence of the different molecules that are in your saliva. And then that can be transmitted along whichever cranial nerve supplies that region. If it's the anterior 2/3 of the tongue, it's conveyed on facial nerve. If it's the posterior third of the tongue, it's conveyed on glossopharyngeal. If it's in the what's called the laryngopharynx, close, and on the epiglottis, then it's conveyed on vagus nerve. Okay, then we go to general senses. All right. Here we have a list of different types of senses, and we're going to identify them as either general or special. I personally just suggest, at least initially, is just write G or S next to them, to the left-hand side of each of these terms, and then you can rewrite them all, these are all the general receptors on the line provided there and all the special receptors. So the free nerve ending is a general receptor. Vision is a special receptor. Meissner's corpuscles, a general. Equilibrium is special. Smell or olfaction is special. Pacinian corpuscles are general. And hearing or auditory is special. Okay, so that takes us to number 8. Receptors can be classified by not only the type of stimulus they respond to, but also where they're located. You can take all these receptors and you can kind of bulk them into, are they exteroreceptors, interoreceptors, or proprioceptors? Because those are the basic categories. Are they, really on the surface of the body to detect changes outside of the body? Deep within the body, let's say along the digestive system, or in the carotid arteries or vagus nerve, where you have baroreceptors and chemoreceptors that respond to the blood pressure being exerted in those vessels or the blood gas levels, right? Those would be chemoreceptors. Those would be interoreceptors. And then we have proprioceptors. Those would be things like muscle spindles, and Golgi tendon organs. So our receptors, can either be classified as where they're located or what they respond to. So that's what we're asking you here, classified by their function. In the space provided, describe the sensation each receptor detects. Okay, so, you can put an M, P, C, or N next to these and then write them out, or you can list them, each, on the line provided. And in this case, I just put them on the line provided. The mechanoreceptors would be the pressure and touch. Those are essentially describing light touch, deep touch, or light pressure, deep pressure, on the surface of the body. Those would be the Meissner's and Pacinian corpuscles. Other mechanoreceptors are associated with hearing, which we'll find in the cochlea, and equilibrium in the semicircular canals and the vestibule. Photoreceptors respond to light. Chemoreceptors, taste and smell. So that's the gustatory cells and the olfactory cells. And nociceptors, pain. Okay, then here we have test your knowledge sensory pathways. Pardon me. All right, so what we have here is, and it's kind of loose because some of these things like pain, touch, and pressure, it depends on where you're talking about. Are these visceral organs, or are we talking about the surface of the face, is going to determine which cranial nerve is going to convey that, because we're actually just looking at cranial nerves here. Spinal nerves are different. And it's just an exercise to kind of really help you organize, some of these senses. What's the receptor type? What cranial nerve have you learned so far that would convey that? Maybe the particular functional cortex area and lobe associated with those, if there is one. All right, so we have the lobe and then a particular functional area, if applicable. Notice this is on the top here, cerebral cortex lobe or brain structure. All right, so one of these, I'll just tell you, equilibrium, is going to be actually received and processed in the cerebellum. Okay, so let's take the first one. We have taste. You know, the type of receptor is a chemoreceptor. The cranial nerve, it depends, right? It could be 7, 9, or 10, facial, glossopharyngeal, and vagus, depending on the location. So I write all three. The lobe, technically it's the insular lobe. However, on the models, we can't really point to the insular lobe. So frequently we'll just point to the parietal lobe, and the functional area would be the gustatory area. Because we have the limitations of that model, I generally don't ask you the lobe, but I could fairly ask you the gustatory area because that's the functional cortex area and the cranial nerves. I could also ask you now, what is the structure that detects taste? That would be a taste bud, or maybe the cells, gustatory cells, or again, the type of receptor would be chemo. On the far right-hand side, I used to have a column there, for some other models associated with it, but I just took it out. So I think this is complex as it is. All right, smell. Receptor type is also chemo. Cranial nerve is olfactory. The lobe is temporal, and it's processed on the olfactory area. Vision, photoreceptor, optic nerve, occipital lobe, visual area. Hearing, mechanoreceptor, cranial nerve, vestibulocochlear, lobe is temporal, and the area is the auditory area. Equilibrium, mechanoreceptor, cranial nerve is vestibulocochlear nerve, because it's the vestibular portion of it. Cerebellum. All right, so the brain structure would be the cerebellum. It's shared with other areas of the brain, of course, but it's going to go to the cerebellum. And so therefore, just put an X in the functional cortex area because it doesn't go to the cortex. It doesn't go to the cerebrum, it goes to the cerebellum. So put an X right there, or just NA. Okay, pain, receptor type is a nociceptor. Now, depending on where it's coming from, and this again, I'm just going to give this to you. I'm not going to ask you the cranial nerve for this unless I say specifically the face. Right? So if it's from the face, right, that would be trigeminal nerve. But if it is from visceral organs, it would be conveyed on vagus. Remember, vagus is the only nerve that provides sensory or motor, to structures below the shoulders and the thoracic, to visceral organs in the thoracic and abdominal pelvic region. The lobe would be parietal, and the functional area would be the somatosensory area. Touch and pressure is a mechanoreceptor. Cranial nerve, again, if it's touch and pressure on the face, then it would be trigeminal. If it's well, you're not going to have touch or pressure in the visceral organs, but pressure, then it would be vagus nerve. And again, parietal lobe, somatosensory area, because it's just general sensation. Okay, so I'm going to stop my share, and I'm going to switch now to the actual course page. So let me switch over to this. And this thing keeps popping up. It's not fun. Oops. Oh, we want to close that out. Thank you. Okay, and close that out again. All right, so let me go to the top of the page. I'm going to go back to the Lab 12 region. So I'm going to switch. I'm going to reshare. Hold on. Because something's not working right. Okay. So here we are in Lab 12 General and Special Senses. We just went through the lab, the PDF document. Right. So now we're going to go through these images. Right. And then here, this is where I have the eye anatomy. So I cover the eye model, extrinsic muscles of the eye, the retinal cell layers and the tunics of the eye, ear, cochlea, and so forth. And then the quiz is, of course, down on the bottom. I did those when we were right at the beginning of the pandemic, when we were being essentially locked out of the building, and I quick ran down there and was able to take some videos on my phone. So they're a little, rustic, but it covers the information adequately. Okay, so here we have the eye model. All right, so we have 1, 2, 3, and 4. And I know you have to kind of swip it around. I don't know what happened to these images when they were uploaded. They just don't seem to be able to be corrected in their location to get them upright. But if you print off some of these, because you can download them, I believe, if you print them off, you can turn it in whatever direction you want. Okay, so we have 1, 2, 3, and 4 on the eye, and 1 is the sclera, 2 is the lacrimal gland, 3 is pointing to the tendon, of the superior oblique muscle. So that's what we'll be pointing to, and the superior rectus muscle. Not that you can't read these yourself, but if I can think of an extra question to apply here, I'll let you know. All right, and here we have another eye model. We have 1, which is going to be lateral rectus. 2 is the inferior oblique, is pointing to the red fibers that are kind of in the shape of a hammock there. They're not fibers, they're going straight to the back of the eye. And 3 is pointing to the optic nerve. So something I might be able to ask you, for instance, is where number 1 is identify that structure or that muscle, and you would say lateral rectus. What if I pointed to 1 and identify the nerve supply, the cranial nerve that supplies, or the innervation, right? That would be lateral rectus. All right, so lateral rectus, inferior oblique, and the optic nerve. Okay, here we're looking at, we take off kind of the upper hemisphere of the eye. We can actually see now the what I call the muscle belly, of medial rectus. Right? It continues anteriorly, of course. Above that is the muscle belly of the superior oblique muscle. All right, so we have the internal region of the eye. 2 is pointing to the inner surface once the vitreous humor is all gone, and that's the retina. 3 is pointing to this region. I believe he refers to that as the central fovea of the fovea centralis. On some models that have the yellow interior, just about where the 3 is located, between the 3 and the tip of that arrow, is a yellow region, number 32, and that's the central fovea, the fovea centralis. But on this model, it didn't have that. It's an older model. So he was pointing to that region for the central fovea. And then 4 is pointing to that red region here, and it looks like muscle, right? So that is the ciliary body that has muscle that acts on the shape of the lens and that has cells that produce aqueous humor. So we have medial rectus, the retina. 3 I said to omit, because we're just going to omit that. I'll find a better area for the, in the video, it points to the central fovea better, that yellow region with the number 32 on it. And then 4 is the ciliary body. Okay, so then we have here 1, 2, 3, and 4. All right, 1 is the lens. Right. 2 is pointing to the cornea, the thin clear plastic covering. 3 is pointing to, whoops, my phone I didn't put it on mute. I'm so sorry. Let me swipe down. There we go. Sorry, let me mute that. Oy vey. All right, the iris is the colored portion. The hole through the center would be the pupil. And 4 represents the volume of fluid between the lens and the optic nerve. that's in contact with the retina in the posterior chamber. That's the vitreous humor that doesn't, replenish itself and keeps the retina pushed into place. And it also helps, absorb some light photons. All right, so the lens, the cornea, the iris, and the vitreous humor. If I was going to point to the pupil, I'd have an arrow that's, going right through the hole, so to speak. Okay, then we're going to look at that middle region, the black region here. Some models have it in brown. This one happens to be black. It's between the outer white part of the sclera and the retina. All right, and that's the majority of the volume here. I mean, if you look back where the optic disc would be, all right, that's all black. So that is your choroid. It continues in that vascular layer, that vascular tunic, anteriorly as the red portion there of the ciliary body, and further anteriorly as the iris. All right, so that's the choroid. It may be black or brown, depending on the model that you're looking at. Okay, this is a picture of the slide of the retina. So let me try to move down a little bit. It's not letting me do that. There we go. I had to just use the down arrow. Okay, so to the lower right-hand side is the cornea. I said it kind of choroid. It looks like dried up blood because it's kind of black. If we had backed out a little bit further, even farther to the lower right-hand side, you'd see some stringy connective tissue stuff. That would be, the sclera. So it goes sclera to the cornea, and now I'm moving from, up to the upper left-hand side. These 3 layers of cells, the thicker purple layer, and then you have a narrower purple nuclei layer, and then you have this other layer of cells right on the surface, and then on to the way upper left-hand corner would be all the vitreous humor. So starting from the outside the choroid, the blackish part, that first layer of dark purple nuclei and things there, that's the photoreceptor cell layer. Then the next layer here that I'm pointing to of a thinner layer of purple staining nuclei would be the ganglion cell layer. And then on the surface would be the, I'm sorry, did I say ganglion? That's the bipolar cell layer. Oh my goodness, I think I need more Starbucks. And then the surface layer of cells is the ganglion cell layer. And their axons are what's traveling to the optic disc to leave as the optic nerves, and then transport these visual impulses to the occipital lobe, to the visual area. All right, so again, from lower right, to the upper left corner, I could probably stop now, right? It's choroid, photoreceptor cell layer, bipolar cell layer, ganglion cell layer. All right, so here, if you could see it, you'd have the sclera to the outside, the choroid, photoreceptors, bipolar cells, ganglion cell. Okay, then we're going to look at the ear models. So we have 1, 2, and 3, the pinna, the external auditory canal or meatus, and the eustachian tube or the pharyngotympanic tube. Okay, the pinna, so, and it's, he put that function down on the bottom kind of as a reminder. So again, let me just go back. I could point to number 1 and say, identify that structure, and you'd say pinna. What if I said, identify that function? So okay, that funnels sound waves into the ear or into the tympanic membrane, the external auditory meatus. I could point to 2 and say, identify that structure, external auditory meatus or canal. What is its function? Funnels, conveys sound waves to the tympanic membrane. Has glands there that produce cerumen to, capture different particles and maybe small insects and keep it from hitting your tympanic membrane. And 3 would be the eustachian tube or the pharyngotympanic tube, and it functions to equalize air pressure on both sides of the tympanic membrane. Okay, so we can take from the model, you can take off that tympanic membrane and you get 2 out of 3 of the ear ossicles. So one is pointing to that membrane in white there. That's your tympanic membrane or your eardrum. Right? Then you have the first bone of the three that's actually making contact with it. So that would be the malleus, and then 3 would be the incus. The stapes looks like the stirrup on a horse's saddle, on an English horse saddle, and that's actually on the other side. So it's quite different. So the one that's in contact with the tympanic membrane is the malleus, then the incus, and then the stapes is the third one. Okay, so when we take out this part of the model, because you can take it right out of the big ear there, you can see the stapes, right? And that's number 2. So you see how it's kind of looks like a horse's saddle. I mean, the where you put your foot, the stirrups, especially on an English saddle, because those are metal. One is pointing to those 3 fluid-filled hoops. 3 is actually pointing to what would be the vestibule, but it doesn't have a number on it, and that's why I don't consistently, so I don't use that. But that number 3 is the area, should you have an instructor that wants to ask you about something like that, is your static equilibrium, and 1 is your dynamic equilibrium. 4 is pointing to that snail shell, the cochlea, that contains the hair cells of the organ of Corti, and 5 is pointing to the nerve. Okay, so one of the semicircular canals, dynamic equilibrium. Then we have the stapes. That's the third of the ear ossicles. On the other side of that is the oval window. We have the vestibule that functions in static equilibrium. I won't ask you the vestibule. Then we have the cochlea and the vestibulocochlear nerve. Okay, this large, if we unrolled the cochlea, these are these 3 scala tube systems that we can see. And when you roll it up, it's kind of like a jelly roll. You just roll it up. So we have on the top here, we have the scala vestibuli. 2 is the scala tympani. And the cochlear duct, or what used to be referred to as the scala media, because it's in the middle, would be between number 3 and these hair cells of the organ of Corti. 4 is pointing to the little hairs there, the hair cells of the organ of Corti. And technically, this white portion to the superior aspect, that's the tectorial membrane. It's a rather bony ceiling. And I think in the video, I point to that, because that stays rather stationary. And as waves are produced in the fluid that's within number 1, the scala vestibuli and the scala tympani, the bottom part of these hair cells here, that's on this other flexible membrane, starts to bob up and down. So we call that the whale, and then that tectorial membrane above it is called the tail of the whale. And the whale starts to bob up and down on the water, so to speak, and those hairs hit that tectorial membrane and deform them, bend those hairs, so that becomes physical, activity here. That makes it a mechanoreceptor, and those impulses are transmitted on the little yellow fibers there, the cochlear nerves, and that becomes the cochlear division of the vestibulocochlear nerve, transmitting action potentials associated with hearing. Okay, so we have the cochlea. This is the cross section of the cochlea. Scala vestibuli, scala tympani, vestibular membrane, organ of Corti, the hair cells, and it should be tectorial, IAL, that's okay. The tectorial, I believe, tectorial membrane. But in any event, that's that white little ledge, that little, we call the tail of the whale, where those hairs from the hair cells will hit. Okay, this is the slide of the taste buds. So here we can see that this is the surface of the tongue. You can see stratified squamous epithelium. That should look familiar to you, like the epidermis. You have the stratum basale, that undulating point. But here that we're pointing to, you can see these rather round little structures here that are the taste buds. And if we can magnify them a little bit more with a little clarity, you'd see that they're opening to this region here. You have one side, which the right-hand side of this crevice that the pointer is pointing to, that black arrow, so to speak. On the other side is the other side of the crevice. They both have taste buds that are opening to this crevice region here. Okay, so those are the taste buds. The cells are the gustatory cells. They are chemoreceptors. The cranial nerves are 7, 9, and 10 that convey that sensation. It just depends on whether you're anterior 2/3 of the tongue, posterior third, or the epiglottis. It is processed in the gustatory area. I won't ask you the lobe. Okay, then we have, oh, here, this is the key to go with it. Taste buds, gustatory cells, chemoreceptors, facial, glossopharyngeal, and vagus, the gustatory area. I think that's the end of the pictures. And that takes us to the videos. So, it's gonna stop. I'm gonna stop it right now. Okay, so you can watch those videos. I think they're really helpful. If I get a chance, I mean, that pretty much tells you what you need to know as far as the histological slides, but I'll go back through there, and if I can do another quick video that replaces the one that's there, that was a live Zoom from the lab room. That's, because I had students, getting in on the Zoom from home. It was a live online lab. Then I will try to replace it. And otherwise, I believe we're done. So let me just go back quickly to this region. Let me close that out. Yep, and I believe, yep, that is it. Okay, so if you have any questions, just give me a holler.